Hi everyone. Today I'm going to present to you a webinar which outlines APC's strategies when it comes to end-to-end development of multi-step synthetic routes. APC is a contract research organisation based in Dublin, Ireland and is part of the Medicine Accelerator. APC specialises in the development of both pharmaceutical and biopharmaceutical processes. We have a proven history of enabling pharmaceutical companies across the globe to accelerate the manufacture of their medicine to the market using our data-rich process development ethos. My name is Ciara MacDonald. I'm a Senior Process Development Scientist at APC, specializing in the area of process development and optimization of multi-step synthetic routes. In this webinar, I will briefly present an overview of the small molecule business unit at APC, alongside an overview of the typical types of projects we work on and our approach to multi-step synthetic projects. I will then present a number of case studies which demonstrate what end-to-end process development looks like in practice at APC and the value APC can deliver to our clients. To finish, I will discuss a future focus for our business, which is in the area of high-throughput experimentation and the journey towards self-driving labs. The Small Molecule Business Unit at the APC is a multidisciplinary team of more than 90 scientists, bringing together chemists, chemical engineers, and analytical experts. Over 80% of our team are PhD qualified. Over the past 12 years, we've supported the development of more than 500 medicines, working across all phases, from early preclinical screening right through to post-approval support. This breadth of experience allows us to understand not only how a process works at a specific stage, but how decisions made early on can impact scalability, robustness, and cost later in development. While APC was originally recognized for its expertise in crystallization, our capabilities have expanded to encompass full synthetic route design and integrated process development. From early route scouting through to late-stage process readiness, we work across the complete sequence of unit operations, starting with the initial synthetic transformations and extending through the reaction, workup, isolation, crystallization, and drying of intermediates, culminating in the final API step. Today, we deliver true end-to-end process design with a strong emphasis on building efficient, scalable, and cost-effective processes for our clients. When it comes to process development at APC, the following slides outline the usual types of programs we work on. Typical engagements include route scouting, route industrialization, process optimization, workup and isolation strategy development, process characterization, flow feasibility and optimization, as well as second-generation process design. While APC routinely supports late-stage activities such as process characterization and preparation for regulatory filings, the focus of today's presentation is on early phase development. In particular, we'll be highlighting our Phase 1 and Phase 2 capabilities, shown here in green. In multi-step synthetic development, we support molecules across the full CMC lifecycle using phase-appropriate development strategies. The main focus of today's talk will be our chemistry workflows in the early phases of a molecule and how we apply integrated process chemistry thinking in early development to de-risk scale-up, establish effective control strategies, and deliver synthetic routes that are robust, efficient, and suitable for successful commercialization. Our development philosophy is rooted in process chemistry fundamentals, with early consideration of reaction kinetics, thermodynamics, mass and heat transfer, and their impact on scalability and robustness. Particular emphasis is placed on reagent and solvent selection and the development of well-controlled reproducible unit operations. At these stages, the high-level synthetic route and key intermediates are typically defined, but substantial flexibility remains in both the chemistry and downstream processing. Reaction conditions, quench and workup strategies, phase separations, isolation approaches, and crystallization pathways are actively optimized. We use data-driven approaches including DOE and mechanistic understanding to control impurity formation, establishing robust operating windows and ensure consistent material quality. As mentioned previously, our philosophy aims to deliver the development of robust, scalable processes for multi-step synthetic routes under accelerated development timelines to deliver enhanced process control. These processes must be scalable, ready for tech transfer, and suitable for the future supply of metric tons of the material. It is a requirement for the processes to be robust in terms of yield and impurity control, and they must be economical, sustainable, with a particular emphasis on delivering safe processes. When we talk about developing the best commercial process, one reality we all share is time. We rarely have as much of it as we'd like. In practice, the window we're working within is the period between identification of the medicinal chemistry route and finalization of the commercial route. Within that window lies a highly dimensional design space. There are, in theory, an almost infinite number of experiment iterations that could be explored. Of course, we can't run them all, and that's why it's essential to use the right tools, which will help us understand the reaction design space and focus our efforts where they matter most. By enabling targeted, data-driven experimentation, we can dramatically accelerate process development and fast-track the optimization of a robust, scalable chemical route. Our first case study will focus on route development to enable API manufacturing. In this case, we were challenged with developing a sustainable, cost-effective manufacturing route to reliably deliver API. The route was received from the client with intermediates locked, and there was a requirement to transform the initial route to a robust manufacturing process. This was a 7-step synthesis including 6 discrete chemistries and 25 overall unit operations. This took 9 months with a total of 15 SMEs. In order to deliver on this project, we used the below practical workflow for accelerated process development. Although the client typically defines the synthetic route, they often don't have access to large quantities of intermediate materials. In most cases, readily available starting materials are purchased, and the client may be able to supply some of the final drug product. However, to maintain efficiency, it's critical that we either source or synthesize sufficient quantities of each intermediate in the route ahead of screening activities. Another important early activity is the generation of analytical standards, which enable the timely development of appropriate analytical methods. We begin with high throughput screening of a reaction conditions into reaction workup and optimization, followed by a demonstration batch. Process safety is considered throughout. DSC is used to identify potential thermal hazards, and reaction calorimetry is applied to better understand reaction energetics. Below is an example of the improvements we achieved on one step of the synthetic sequence. This was an amide coupling reaction. The original process suffered from poor conversion, high impurity levels, and a reliance on toxic reagents. By optimizing reagent selection and applying kinetic modeling, we were able to significantly improve reaction performance. Conversion and yield were increased, impurity levels were reduced, and we ultimately achieved a yield of 92%. This was an improvement on the previous 53% yield. Further gains came from streamlining the workup and eliminating a lengthy solvent swap during isolation. As a result, total solvent volume usage was reduced from 40 to just 9 volumes. In parallel, we replaced the original reagent with a greener alternative. Taken together, these improvements delivered a 44% reduction in process mass intensity. At a high level, our strategy enabled the delivery of a sustainable, cost-effective manufacturing route. On the first tile on this slide, we can see how our approach began with yield improvements as previously specified using kinetic modeling, and this allowed us to maximize product formation while minimizing impurities. Additionally, we designed the workup and crystallization steps to reduce losses. Overall, these improvements resulted in a 20% increase in overall yield on the route, which was a significant gain for both efficiency and cost. Sustainability was a key focus, and we reduced the use of undesirable reagents and solvents throughout the route. PMI was tracked across the sequence to identify and build reductions. Cost of goods was further reduced by performing efficient chiral resolutions early in the sequence at step 3. Through careful screening of resolving agents based on the eutectic composition, we achieved 90% yield and greater than 99% enantiomeric access. Importantly, racemization was avoided throughout the remainder of the synthetic sequence. The flow mode enhanced process safety, especially when handling hazardous reagents. It also improved output quality due to superior mixing profiles. Continuous processes increased overall efficiency. Telescoping of two steps enabled the removal of a total of 6 unit operations from the manufacturing sequence. Finally, the optimized route was successfully transferred to manufacturing scale. This enabled our client to safely deliver high-quality medicine to patients, achieving both efficiency and sustainability goals. This project exemplifies how thoughtful process design can deliver both sustainable and cost-effective manufacturing without compromising quality. Our second case study will focus on a route feasibility assessment followed by optimization for an RSM for a client. The design requirements from the client was that they required greater than 70% yield from the route, low impurity levels, good mass balance understanding, alongside a preference to synthesize this RSM in flow. The synthesis of this molecule is not documented in the chemical databases. Therefore, the first step of this program was route selection, whereby the chemical databases were used to identify 10 potential routes to the RSM. The chemical databases were used to find precedent for the selected chemical transformations and the potential reaction conditions that could be employed. Following on from this paper-based exercise, 3 routes were selected based on known synthetic pitfalls, freedom to operate, commercial viability, safety, and sustainability. Once the 3 routes had been selected, we entered the route evaluation phase, whereby data-rich first-pass experiments supported by PAT were performed using the literature conditions. This enabled the collection of key analytics and learnings on the processes. The resulting reaction mixtures were then purified via our automated chromatography purification platform, which includes both a PrepLC and a biotage system. This step enabled the determination of isolated yields from each route and additionally characterization of the main products and major impurities of each reaction. A combination of NMR and LCMS is used to confirm that the desired product had been formed. Based on this initial evaluation, the selected routes were reprioritized as per the last box on my slide. This was based on output yield, selectivity, crude purity, and workup requirements. Additionally, HPLC method development was able to commence at this stage to support the screening and optimization stages. In this case study, only 2 routes were progressed after the initial evaluation phase due to issues with chemoselectivity. For the remaining 2 routes, we moved into the screening and optimization phase. An experimental plan was devised combining the scientific literature and digital tools, for example, pKa estimation in order to select suitable bases. A vial screening approach was used on the 2 remaining routes to screen the following parameters: reagent, base, catalyst, solvent, and stoichiometry. These results were used to lock solvents and reagents. At this stage, based on cost of goods, yields, impurities, downstream implications, and adaptability to flow, 1 route was selected for further optimization. Initially, with the reagents and solvents locked, our Ceres Asia flow system was used to optimize the reaction with respect to temperature and residence time. In the final optimization phase, it was found that greater than 90% yield could be achieved in batch through the control of the rate of addition of reagents. Additionally, the use of a cross T-piece inflow enabled simultaneous introduction of reagents, and this allowed for a greater than 90% yield from the reaction. With feasibility and optimization to 90% yield performed on this route, it was successfully transferred to the client site. Impurities were well understood and purged through the workup. PAT had been identified for reaction tracking alongside the development of 5 key HPLC, GC, and chiral analytical methods. Finally, the reaction was successfully adapted to flow. The final case study will highlight a future focus at APC. This is our high-throughput experimentation journey towards self-driving labs. We hope to combine flow chemistry, machine learning, and automation to establish the research platform of the future. In order to achieve this, we are using the Ceres Asia flow chemistry system, which allows pre-programmable reaction conditions and automated HPLC sampling alongside integrated FTIR. The reactor platform, including its pumps and modules, are controlled externally. Communication between the reactor and this external control system would enable reaction optimization through the use of FTIR data to influence reaction conditions, for example, stoichiometry and temperature. To begin with, we started with a proof of concept experiment whereby the system used Bayesian optimization in combination with FTIR data to optimize the concentration of sodium hydrogen carbonate in the system by modulating flow rates. The system was found to successfully optimize the concentration of sodium hydrogen carbonate after 10 rounds of self-optimization. The optimum conditions were identified after 7 rounds. As a proof of concept, this successfully demonstrates the system's capabilities. Our next step is to apply this to a real-world chemistry problem and see if the same system can be used to successfully optimize a reaction by self-optimizing the concentration of the product of the reaction using FTIR data. And we're excited to see where this experimentation leads. Thank you for listening to our webinar today. If you would like to find out more information on APC and are interested in working with us, please visit approcess.com or visit our LinkedIn.